Hi everyone, my name is Pathik, and today with my colleagues Shruthi and Isaac, we will be discussing a paper regarding the segregation of emotional memories within the hippocampus. First off, let's have a quick overview on the hippocampus and its roles. As we all know, the hippocampus is a brain structure found deep in the temporal lobe of the brain. This structure plays an important function for learning, spatial navigation, and for the purposes of this paper, memory. Specifically, the hippocampus controls emotion memories, which can have a positive or negative valence, meaning they can be associated with good emotions or bad emotions. Previous studies in the hippocampus have suggested the ventral hippocampus selectively segregates and relays information regarding emotional memories based on valence. But how multiple engrams of varying valences are represented within the same brain region remains poorly understood. Now you may be wondering what an engram is. And that is important to understand as this paper makes references to engrams, which are an important construct for memory. Simply put, engrams are a set of neurons activated together across various brain regions corresponding to a given memory. When that specific pattern of neurons are activated together, we can retrieve a memory, and as such, engrams are considered a physical trace of memories. Now that we know some key terms and background information, let's discuss the objective of the paper. This study aimed to identify how appetitive or positive and aversive or negative memories are differentially stored in engrams within the CA1 cells of the ventral hippocampus. This objective was carried out using a mouse model. There were three key methods that were used to explore this objective, including tagging, genetic differences, and behavioral differences. First off, the CA1 cells of the ventral hippocampus were tagged to measure overlap of projection patterns within appetitive and aversive engrams. Next, a genome-wide RNA and DNA methylation sequencing was done to investigate genetic differences between the neurons responding to appetitive and aversive stimuli. Finally, the subjects were behaviorally tested to determine the functional flexibility of ventral hippocampal cells. Now, let's explore some specifics regarding the methods. To go over some of the general protocol, this study was conducted on male transgenic TRAP2 mice as well as controls. There was immunohistochemistry carried out, which is a method for visualizing tissue. This was done using two CFOS-based tagging methods to visualize engrams. The mice were placed in groups based on which behavioral trial they participated in, with a general diet that was followed. The mice were placed into four groups: aversive appetitive restraint, aversive restraint, appetitive aversive condensed milk, and appetitive condensed milk. Each subject was given a diet of food plus doxycycline 48 hours prior to surgery. And this surgery was needed for optogenetics. After a stereotaxic surgery and optic implant, the mice were given 10 days minimum to recover and were given a standard food diet. After this, the behavioral trials took place with an appetitive, aversive, and CFOS-based third trial, which was either restraint stress as an aversive trial or condensed milk as an appetitive trial. The stereotaxic surgery mentioned earlier involved bilateral craniotomies, which means small holes were drilled into the skulls of mice. Bilateral optic fibers were placed above the injection site, and the mice were injected with a cocktail of viruses with 300 nanoliters of cocktail per site at a rate of 100 microliters per minute. In order to tag appetitive behavior, female exposure was done, as this is considered a positive experience for the mice, and this was tagged using EYFP. Similarly, the mice were given foot shocks as an aversive stimuli, and this was tagged using TD tomato. For the aversive trial, mice were placed in a shock chamber with light stimulation for 500 seconds, and they were given four foot shocks over these 8 minutes. The mice were also given 4 OHT on the day of tagging, which is important for tagging, as mice that were injected with DMSO instead of 4 OHT showed no TD tomato expression. To acclimate these animals to injections, they were injected at least twice beforehand with saline or DMSO. For the behavioral assay, the mice were given 10 minutes to explore the arena, and this was repeated if the mice showed a preference for either side of their cage. As mentioned earlier, there was an optogenetic induction involving female exposure as an appetitive behavior, as well as foot shocks as an aversive behavior. In order for immunohistochemistry to be conducted, the animals had to be sacrificed with their brains extracted and 50 coronal sections made. This allowed the tissue to be visualized. Cells were also quantified for animals with accurate bilateral injections using a confocal microscope. The number of EYFP, TD tomato and CFOS immunoreactive neurons in ventral CA1 cells were counted to measure the number of active cells with about 3 to 5 coronal slices per subject. Finally, a single cell suspension was made from the mouse hippocampal tissue with the brains extracted and the region isolated. Additionally, an RRBS sequence library was prepared and analyzed to measure genetic differences between cells shown to be important for appetitive versus aversive engrams. This sequencing was done on tag nuclei, which were sorted using FACS, and cytosines that were covered by at least 5 reads were used for analysis, since those residues would be methylated, and this study partially focused on DNA methylation patterns. There were 494 DEGs, or differentially expressed genes, between the groups, and gene ontology, or GO analysis, of the up and down regulated pathways was done in aversive CA1 cells.
So now onto the results of the study. One of the first things that they found was that there was a distinct anatomical segregation of the cells that were recruited for appetitive and aversive engrams. Using a dual tagging system, they found that the appetitive engrams, which had the first tag, EYFP, were mostly found in the posterior VCA1, while the aversive engrams, which had the second tag, TD tomato, were mostly found in the anterior VCA1. As an extension, when the cell populations were reassessed upon subsequent exposure to experiences of a similar valence, they found that there is a high rate of co-localization of the tags. This can be seen in the first 3 figures here, where either 2 appetitive or 2 aversive experiences led to higher rates of co-localization than 2 experiences of different valence. In figure J and K, stress represents aversive, while milk represents an appetitive experience. Figure L shows that the 3 aversive experiences, group 2, or 3 appetitive experiences, group 4, also has really high co-localization, indicating that the same cells are being recruited for aversive or appetitive experiences. Using dual tagging again, the brain regions involved and the outputs of these cells were studied. There was clear structural segregation and heterogeneity of the projections from these cells. As observed by fluorescence intensities, appetitive projections mostly went to the basomedial amygdala posterior part, the BMP, the anterior commissure anterior part, ACA, the fornix, medial hypothalamic nucleus, and the lower layer of the dentate gyrus, while aversive projections mostly went to the anterior BLA, the posterior BLA, the NAC core, and the upper layer of the dentate gyrus. Next, RNA sequencing was used to assess the molecular profile of these VCA1 cells. This showed that in total, aversive VCA1 cells had 474 differentially expressed genes, or DEGs, compared to the neutral group, and appetitive cells had 1,104 DEGs compared to the neutral group. Among these, only 262 DEGs overlapped between aversive and appetitive cells, showing that there's a unique and distinct transcriptional landscape in these 2 populations. Furthermore, the top 20 downregulated DEGs showed no overlap with each other, again supporting the distinct transcriptome in these neurons. After this, gene ontology analysis was completed to look at the molecular signatures of the transcriptome. The top upregulated pathway in both aversive and appetitive engrams involves neurotransmitter complexes, such as ionotropic glutamate receptor activity, and the top downregulated pathway was different between the 2 cells. So aversive cells downregulated DNA mismatch repair, while appetitive cells downregulated axoneme assembly and microtubule bundle formation. The epigenetic landscape was next explored using reduced representation bisulfite sequencing, or RBBS, as epigenetic regulation is known to occur in engrams. Specifically, researchers looked at the DNA methylation landscape and found that they are different between aversive and appetitive cells. When looking at differentially methylated genes, DMGs, which contains at least 2 differentially methylated cytosines, only 32 DMGs are shared. The top 20 DMGs in each cell population also show no overlap between each other, thus showing the distinct DNA methylation landscape between the two populations. Next, to see whether there is a causal role of the tagged VHPC cell bodies in driving behavior, an optogenetic induction protocol was used. Stimulation of VCA1-BLA or VCA1-NAC terminals was found to drive aversion. When these mice received an appetitive experience while receiving optical stimulation, post-induction, the cells had found to switch their drive. Specifically, VCA1-BLA now drove preference or appetitiveness, and VCA1-NAC had reset to baseline levels. Similarly, optical stimulation of VCA1-BLA and VCA1-NAC was sufficient to drive place preference, but when induced with an aversive experience, the cells had now switched. Now, the VCA1-BLA had switched to driving aversion, while VCA1-NAC had reset to baseline. Altogether, this showed that the VHPC cells relayed their behaviorally relevant and valence-specific content to downstream targets, despite sharing partially non-overlapping molecular and neuronal signatures and distinct projection patterns.
And finally, for the discussion, we'll first present some general conclusions, then some alternative interpretations for our data, and finally, some limitations and future steps. So from the results, the researchers concluded that there's two regions in the ventral hippocampus that process appetitive and aversive behavior. And we know that these regions are distinct on a cellular level. Again, we're reminded of this by the left diagram where appetitive cells staining green and aversive in red don't overlap. They're distinct on a transcriptional level where we learn that there is different genes being downregulated between appetitive and aversive events. And they had different projections. On the graph on the left, we see a lot of TD tomato fluorescence in the projections to the anterior basolateral amygdala, indicating that aversive cells project to this brain region. And it's these different projections that help to optimize how emotions are integrated for memory. So why are these results significant? Well, past papers were able to determine that certain brain regions respond to specific events. For example, a 2016 paper by Kim and colleagues found that positive and negative stimuli converge on the basolateral amygdala. Likewise, a 2019 paper found that the prelimbic cortex controls fear. However, in these papers, it was unclear how exactly these behaviors are represented in said brain regions. In our paper, the cellular and transcriptional differences help to show how specifically these responses, which respond to behaviors of different valences, are represented in the ventral hippocampus. And again, the appetitive cells are located in the posterior region and aversive in the anterior. As for projection differences, they help to provide insight on the behavioral component of the ventral hippocampus. In a previous paper by Su and colleagues, they found two distinct pathways from the ventral hippocampus to the amygdala to modulate fear. And in this paper, the researchers found 11 additional projections to various brain regions, and they found that these projections are plastic, meaning they're subject to change based on what behaviors they are exposed to. Speaking of plasticity, this leads us to our first alternative interpretation of the paper, which is the idea that the VCA1 cells in the hippocampus, so the cells that were looked into for this study, that they're hardwired to process appetitive or aversive behavior. Now, the researchers argue that it's not so much these cells start off processing appetitive or aversive behavior, but that they become that way after exposure to such events. And we have reason to believe this by looking at the two diagrams on the bottom, which show projections of VCA1 cells to the basolateral amygdala and nucleus accumbens on the right. First, in the pre-induction period, the mice were subject to aversive behavior, and we see some projection, although less than baseline, as indicated by the orange bars. Immediately after, the same mice were moved to cages where they were then exposed to appetitive behavior. And we see those projections go up to baseline or even significantly higher, as indicated by the blue bars. This tells us that the same projections can respond to appetitive and aversive behavior. So rather than being hardwired for either, there's flexibility to change what they can respond to. Another interpretation of the data is that we saw different cells being activated due to fundamental differences in stimuli, and this would have led to different genes and anatomy being involved. And so to compare the two chosen stimuli, social interaction and fear, we first see that they're processed in different areas of the brain. And even in nature, social interaction is more multimodal with social and context cues, whereas fear is more unimodal. In this case, a foot shock would have caused a very specific anatomic response. And so fundamental differences in the nature and area of processing could be a reason why we saw two populations of cells being activated in the hippocampus. As for limitations, the first is that the researchers only tested on male mice. And we know that behaviors such as social interaction is sexually dimorphic, meaning it differs between males and females. Another limitation is the tagging timeframe. The mice were allowed to, or they were exposed to EYFP for 48 hours and TD tomato for 72. This is way beyond the expression time for both tags, which take about 1 to 4 hours, and this could have led to the overtagging of cells that are not directly involved in processing appetitive or aversive behavior. Finally, CFOS as an expression marker can be a limitation. CFOS is expressed immediately following neural activation, so it is good at targeting immediate responses, but it's not so great at representing slower timescale events like changes to gene expression. So where do we go from here? Future studies can first look into characterizing the cells in the ventral hippocampus. We learned from the study that the cells are different for different valences, but are cells for the same valence different, or are they the same? For instance, in the basolateral amygdala, we know that the response depends on which region is activated, and perhaps that's the same in the hippocampus. For instance, we know that the anterior portion responds to aversive events, but maybe activation of different portions in the anterior axis can lead to different aversive responses. Future studies can also conduct the experiment using a unimodal stimulus for both behaviors. For example, instead of female-male interaction, they can look into using vibrations, which is similar in nature to foot shocks. And finally, studies can observe axonal outputs over time, looking at factors like dendritic growth and retraction, or changes in axonal interaction to better understand the plasticity of these ventral hippocampal projections. These are some of the references we used. And that concludes it for our presentation. We'd be happy to answer any questions you guys had for us.